join. I hope that recording the session wouldn't be an issue with you, Prof. Amiji. Do you have any?
No,
Um,
yeah, no, that's
yeah,
fine. Yeah,
because usually we keep the recordings available in our web just to others to come back and have a look. Yeah.
yeah, no.
Okay. Thank you very much, Prof. Amiji. So I have the honor of introducing our today's speaker, Professor Mansoor Amiji, who is currently the University Distinguished Professor and Professor of Pharmaceutical Sciences and Professor of Chemical Engineering at Northeastern University in Boston, USA. Professor Amiji received his bachelor's degree in pharmacy from Northeastern University in 1988, and then he pursued his PhD in pharmaceutical sciences at Purdue University and obtained the PhD in 1992. Professor Amiji is teaching at both undergraduate and graduate student level at the Northeastern University right now, and his research is focused on development of biocompatible materials from natural as well as synthetic polymers, also targeting specific drug and gene delivery systems for cancer and infectious diseases. And also he's working on nanotechnology application for medical diagnosis, imaging, and therapy as well. Professor Amiji has edited over 12 books and has published over 60 book chapters. And moreover, he has over 350 peer-reviewed publications, which earn him an H-index of 103. So he's one of the highly cited researchers in pharmacology and toxicology area, which is coming under top 1% of the scientists. Moreover, Dr. Amiji has received a number of honors and awards, including the Nanoscience and Technology Institute Award for outstanding contributions towards the advancement of nanotechnology, microtechnology, and biotechnology, and then American Association of Pharmaceutical Scientists Meritorious Manuscript Award, also a Control Release Society's Nagai Award, and currently he is an elected fellow of both the American Association of Pharmaceutical Scientists and Control Release Society of USA. So he has an impeccable track record in his area of research. So we are glad to have you here today. And today, Professor Amiji will be talking to us about extracellular vesicles, mediated communication in the tumor microenvironment. So the floor is yours, Professor Amiji.
Thank you. That's a very nice introduction, and appreciate the kind words. Good morning from Boston. Good afternoon for you in Estonia, and thank you so much wherever you're joining us from. I know this invite was extended to other individuals who may be joining from other parts of the world. So it gives me great pleasure to be here and to tell you a little bit about our work on looking at extracellular vesicles as mediators of communication. We are interested in extracellular vesicles or exosomes. Again, the terminology and the nomenclature is there's a lot of debate about what are exosomes versus what are extracellular vesicles. But I think generally speaking, I'm going to use the term extracellular vesicle just to denote in a more larger context, as opposed to anything that is more characterized as exosomes. But what I wanted to talk to you today was really focusing on tumor, and specifically in the context of cancer. How can EVs help us in understanding the tumor biology and ultimately create better therapeutic strategy? So in 2000, first, a seminal paper was published by Doug Hanahan and Bob Weinberg, that was called The Hallmarks of Cancer, and they identified several of these hallmarks specific to cancer cells that are different from normal healthy cells. And from there, they basically were interested in characterizing these hallmarks. And then in 2011, they published a second round of that paper, which one of those figures is cited here. and they increase the number of these hallmarks. And you see, for example, tumors have these properties of evading growth suppressors, or have this immortality, promote inflammation, angiogenesis, or formation of blood vessels, resisting cell death or apoptosis. And more, we believe that these hallmarks are not necessarily just a property of the cancer cell itself. It is actually the connections between cancer cell and other cells that live in that microenvironment, and how those interactions and messages that are transferred from one cell to another is really creating these types of hallmarks, these unique features that are present in tumors throughout the body, irrespective of their site, whether they're ovarian cancer or breast cancer or pancreatic cancer. And one of the areas that we are very intrigued about is the communication between cancer cells and immune cells that infiltrate into the tumor. And you'll see evidence from our work, specifically focusing on myeloid-derived cells that infiltrate into the tumors and subsequently have very interesting properties in terms of the biology of the tumor. So in summary, from here, the main lesson that we and others are focusing on is, what is this relationship between a cancer cell and neighbors, whether they're endothelial cells, immune cells, fibroblasts, or other types of cells that are present in the microenvironment. How is that communication modulated? How can we interfere with that communication in order to then enhance therapeutic benefits and help treat these cancers in a much more favorable way? So as I mentioned, EVs are key mediators of that communication, and to this audience, I don't have to go too deep into the discussion around extracellular vesicles. These are endosome-derived luminal vesicles, and previously they were thought to be simply garbage bags that the cells were excreting whatever material that was not needed in these exosomes, and then subsequently they were used mostly for excretion purposes. However, we are realizing now the role of EVs and how EVs are such important mediators of communication from the donor cell to the recipient cell, and how they control the message in a very, very specific and selective manner. Just in terms of the properties of EVs, from 30 to 100 nanometer in diameter. We and others utilize mostly ultracentrifugation and tangential flow filtration and gel filtration methods to isolate them. And then we characterize both the surface properties of EVs, specifically looking for various types of markers on the surface, as well as most importantly, the content of EV. And one particular content that we are interested in, I'm going to spend probably most of the time today, is to talk about microRNAs. These are non-coding RNAs that are produced by tumors and other cells, and ultimately are secreted in extracellular vesicles. These microRNAs then are transferred over to the recipient cells, and the recipient cells can then have specific properties and have alteration of their phenotype based on these microRNAs. And so we and others have been very interested in asking questions related to these microRNAs. What are these microRNAs? What are specific microRNAs that are produced? What can we do to change the content of these EVs in order to change that content of microRNAs? And can we specifically artificially include certain microRNAs in EVs that would have a therapeutic effect? And I hope I can convince you with several examples that that's a possibility. So EVs have, not just in terms of properties of regulating this communication, but through that regulation of communication, they're able to have significant effect on angiogenesis and metastases, promote tumor growth and proliferation, suppress tumor immunity, promote resistance to anti-cancer therapy, as well as modulate metabolism. As we all know, tumors generally metabolize glucose through aerobic glycolysis, and a lot of these properties are mediated through extracellular vesicles and the content of these extracellular vesicles as they are transferred from the donor cell to the recipient cell. My focus today is going to be mostly around two areas. One is the question of resistance to anti-cancer therapy, which I'll talk a little bit later on. But in the beginning of my presentation, I'm going to focus mostly around the role of EVs in terms of communication in the tumor microenvironment and suppressing immunity, and ultimately creating this immunosuppressive microenvironment. And then how can we artificially look at certain microRNAs in these EVs and see if we can change the properties of the immune cells, specifically myeloid cells, in the tumor microenvironment to create a more favorable anti-tumor response. So in that discussion, we have been focusing on these tumor-associated macrophages, or these myeloid cells. And just to give you a background, there are two types of macrophages that are present in all of our tissues. We have what are called resident macrophages that are obtained through embryonic origin, and then we have infiltrating macrophages derived from bone marrow and ultimately travel through the bloodstream and come into different areas of our body. And generally, it's known that in tumors, once tumors start to grow, depending on which organ systems is involved, you will have certain types of resident macrophages. But majority of these tumor-associated macrophages that are getting into the tumor and ultimately creating this microenvironment are really infiltrating myeloid cells. They come in through monocyte-derived monocytes, and then subsequently monocytes are transformed into macrophages based on the microenvironment that is present in that tumor. And then ultimately these macrophages acquire a phenotype, which is called tumor-associated macrophages. And these are the ones that tend to be very important in controlling the homeostasis of the tumor, in allowing it to propagate, allowing angiogenesis to occur, resistance to develop, and so forth. So these infiltrating macrophages, once they enter the tumor microenvironment, they acquire a specific tumor-associated macrophage phenotype. And then subsequently, this tumor-associated macrophage phenotype, which is currently recognized mostly as having an M2-like phenotype. So these macrophages, they are in a spectrum of phenotypic properties, and generally the ends of those spectrums are characterized as M1 being the anti-tumoral, pro-inflammatory, and M2 being the pro-tumoral, pro-fibrotic phenotype. And the questions that we and others have been asking is, if these myeloid cells in the tumor, these are tumor-associated macrophages, acquire this specific property, is there a way to engineer them to change their phenotype and ultimately create this pro-inflammatory, excuse me, but anti-tumoral or M1-like property? So one of the markers, the hallmark of tumor-associated macrophages, is overexpression of a receptor on the surface called CD206. This is also called sometimes Mannose receptor, and this receptor is, as you'll see in a lot of the data that I'll present, is what is present in the tumor and allows the tumor to propagate. As you see from this figure here, back in 2011, there was published that showed that through this tumor-associated macrophage phenotype, you have angiogenesis, you have cell migration, invasion, metastasis, suppression of the immune system, as well as all of the other hallmarks of tumors that make it more aggressive. So our idea is, how do you control the message between tumor cells and tumor-associated macrophages? What causes these to acquire specifically an M2-like phenotype? And then subsequently, how can we change that phenotype through exogenous administration and transfection of various types of microRNAs, and allow those microRNAs to be secreted in EVs, and then have those EVs enter the tumor-associated macrophage. So the tumor cells become the donor cells, tumor-associated macrophages become the recipient cells. And the question that we've been asking for many years now is, are we able to control that phenotype, and can we create an pro-inflammatory anti-tumoral phenotype of these macrophages? So again, our focus has been mostly with microRNAs. As I mentioned, these are non-coding RNAs present in all of our cell types, and they are master regulators of the cellular phenotype. And within those M1, M2 phenotypes, as I was discussing, there are many different types of specific markers that we can look at, cell surface markers like CD206, CD81, and so forth, which are present. CD206 is present on M2, CD81 is present on M1. We can also look at various ways to stimulate these cells. For example, if you stimulate macrophages with LPS, you induce M1 phenotype. If you stimulate with IL-10 or TGF-beta, you are basically converting them towards M2 phenotype. And these characteristics are well defined now that you can do flow cytometry and see the specific hallmarks that would be characterized by whether M1 or M2. And literature has also shown us that certain microRNAs, if you have an M0, which is your baseline phenotype of macrophages, if you stimulate it with microRNA 125B, for example, it will convert it into an M1-like or a pro-inflammatory anti-tumoral phenotype. Similarly, microRNA 155 does the same thing, and you'll see a lot of data both from 125B and 155. However, if you want to switch to an M2 or more pro-fibrotic wound healing phenotype, you would basically have to transfect these cells with microRNA 223 and ultimately show that you are able to change the phenotype from M0 to M2. So we know these microRNAs are present in all of our these different types of macrophages, especially tumor-associated macrophages. And our goal is to convert them from this pro-tumoral phenotype to an anti-tumoral phenotype, and then look to see if EVs can help us in terms of delivery of these microRNAs from the tumor cell to these myeloid cells and ultimately change that microenvironmental phenotype. So the data I'll show you today mostly will focus on microRNA 155 and 125B, but we have also done the reverse, which is transfect myeloid cells with microRNA 223 and convert them into a pro-fibrotic, pro-wound healing phenotype in the context of inflammatory disease and wound healing. This work in my lab has been mostly funded for the last 15 years now through the NCI, National Institutes of Health, National Cancer Institute here in the United States. So to deliver these microRNAs to cancer cells, I had some really talented students and postdocs in my lab. Ashanti is now at a company called Dicerna here in Boston, Cambridge. She is an associate director. Arun Iyer was a postdoc in my lab. He's now an associate professor in Detroit in Wayne State University. And Amit Singh was also a postdoc in my lab. And he is now heading a cell and gene therapy program at Takeda. And they came and we asked this question: could we create a platform technology that allows us a non-viral way to deliver nucleic acid to different types of cells? And so our idea was to create a system where we can encapsulate nucleic acids, we can create nanoparticles, use safe materials with a history of clinical applications, such as hyaluronic acid or dextran, and then subsequently make these, the chemistry around these conjugates was designed such that it would be simple and straightforward and scalable. And the idea was to use materials either that had intrinsic targeting ability to cancer cells or to myeloid cells, as well as being able to then package the nucleic acid and deliver them to cells and help us in terms of transfection. And so the idea of creating this nanoparticle really came about from how Lego pieces assemble, and ultimately you create a structure based on bringing Lego pieces together with different shapes and different types of structures, such that you have that final property that you desire. And we have encapsulated a number of different types of nucleic acid in these nanoparticles and published a lot over the last 15 years now. But I'm going to focus mostly on today's discussion with microRNAs and how do we take microRNA mimics, which can be then utilized to deliver to these cancer cells and then ultimately use EVs endogenously to try to increase the delivery efficiency to other cells, including myeloid cells. Some of the earlier data, we also have microRNAs that are produced by transfecting with plasmid DNA. So DNA is entrapped in our nanoparticles, and then the DNA then codes for microRNAs in the cell. So you'll see both just microRNA mimics, which are double-stranded nucleic acids with 24 base pairs, similar to siRNA, or a DNA that it then encodes for microRNA in a cell culture or in vivo system. So these hyaluronic acid nanoparticles, for example, that we have made, our starting material is hyaluronic acid with a molecular weight around 10 to 15,000. We then we have the chemists in the lab who can synthesize different functional groups in these hyaluronic acids. So basically create what are called comb-like structures, where you have a backbone of hyaluronic acid and then appendant groups that come out of it, such as a peptide or through a polyethylene glycol spacer, a lipid tail with any carbon chain length from C5 or C4 all the way up to C22. Polyethylenamine is a cationic polymer, and therefore it's important for nucleic acid complexation. We have used low molecular weight PEI, and we have controlled the density in such a way that we are getting sufficient level of cationic charge. Again, remembering the hyaluronic acid is negatively charged, so it doesn't complex with nucleic acid, but putting a cationic charge allows us to complex nucleic acid, but still we retain that backbone structure. We have also put PEG chain, we have put thiol groups in some of our structures to create disulfide crosslinks, and then when these systems get in contact with glutathione in the cell, in the cytoplasm, the disulfide bonds break and release the nucleic acid payload. So we have engineered these types of systems, and then our approach is using a fairly high throughput way to test libraries of these nanoparticles. First, in just to do they encapsulate nucleic acid? Does the particle size is around 50 to 100 nanometer in diameter? Are these nucleic acid molecules stable, and can this structure deliver in the cell? And then we start going into the cells, whether it's tumor cells or macrophages or other types of cells, mammalian cells. We would look to see if the structure is actually allowing the nucleic acid to escape the endosomes and then get into the cytoplasm. Is the payload released in the cytoplasm? Is it bioavailable? Can it actually engage with the cell machinery to affect the properties that we desire? If it's a plasmid DNA, does it go to the nucleus and actually transfect? And then once we start to identify certain structure functional relationships, we can iterate this process and come up with even better structures. But ultimately, we go to in vivo systems and test this usually in rodent models of diseases to see if the hypothesis that we have started with is actually true or not, or does it work or not, and then come up with efficacy and safety data. So these formulations have been well characterized. As you see, this is a story that's about 10 years old now. Was first published in 2013 in a biomaterials article where we designed a number of these HAPEI and HAPEG particles. So this was done with 50-50 ratio of hyaluronic acid conjugated to PEI, very small groups of PEI, what you see as pendant here, and the green here represents hyaluronic acid, and then a polyethylene glycol, and we mix them 50-50 by weight. And what you get is a structure that has a core shell property. So you may be able to see this electron micrograph. The nucleic acid is encapsulated in the core, and the shell is still hyaluronic acid. So it still recognizes CD44 on these different tumor and myeloid cells. And if you put a negatively charged or a counter ion like polyacrylic acid, because it's electrostatically complexing to the positive charge of PEI, the nucleic acid, in this case the siRNA, is released 100%. So it is electrostatically complexing with the nucleic acid and protecting. The encapsulation occurs in the core of the particle. The shell is still HA or hyaluronic acid, and subsequently it still facilitates internalization through the CD44 expression in myeloid cells. So based on these characteristics, we started to ask, can microRNAs be encapsulated? How will microRNA be delivered specifically to myeloid cells? And how can we make sure that it's only the myeloid cells that are taking it up? And ultimately, how can we make sure that we can change the polarity of these myeloid cells towards the anti-tumoral phenotype, and then have it work in the context of specific diseases? So the area that has been a great importance in my lab over the last several years is pancreatic cancer treatment. And pancreatic cancer is a very challenging disease. Right now, worldwide, there are about almost 500,000 cases. And you see that most of the countries of the world, especially where there's a high density of cases, are usually North America, Europe, Australia, and places where right now we are seeing a huge increase in pancreatic cancer, almost correlating with the rise in diabetes. And many folks are thinking that because of this rapid rise in diabetes, type 1 diabetes especially, we are going to see an increase in pancreatic cancer as we go towards the latter part of the century. And the challenge in pancreatic cancer is that because the disease is diagnosed at a very late stage, usually in stage 2 or 3, there's a very, very high mortality rate. Almost you can see how much 100% of patients die because of this disease within 5 years. And one of the challenges in pancreatic cancer treatment is that the tumor forms this very, very dense stroma, what is called desmoplastic stroma, which is almost like a rock in the tumor that prevents drug penetration. It more importantly, even prevents antibodies and some of the immunotherapy penetration. So the question that we've been asking very much is, how do you modulate that phenotype of various cells that are in that tumor microenvironment, especially tumor-associated macrophages, because this has been very important. Macrophages are the ones that secrete procollagen and other growth factors that stimulate and through cancer-associated fibroblasts, stimulate that fibrogenesis and ultimately formation of that dense stroma. So how can we reprogram these macrophages towards an anti-tumoral phenotype and remove this tumor immunosuppressive microenvironment? And ultimately, could we make sure that not only are we improving the anti-tumor immunity, but also decreasing this collagen formation that will help in improving drug therapy, improving permeability of drugs, as well as improving the permeability of antibodies for immunotherapy. So Meiju was a master's student in my lab. She's now working here at Dana-Farber Cancer Center. And the first question we were asked is, how can we look at, specifically in the context of pancreatic cancer, in a cell culture system, the role of extracellular vesicles in communicating with macrophages? So Meiju designed a system where we have transwells. You are probably familiar with, these are inserts that are placed in cell culture, and you can have the top layer here, in our case, is pancreatic cancer cells, human pancreatic cancer cells called PANC1. And the bottom layer, at the bottom here on the in these culture inserts, we put these J7741 alveolar macrophages, and we grow them basically in a monolayer culture. And then we are looking to see the communication between the tumor cells and these macrophages based on different stimulations that we provide to these tumor cells to see how these macrophages respond, and ultimately what happens to these macrophages based on some of the content that is transferred from the pancreatic cancer cells. So initially we stimulate these pancreatic cancer, the macrophages with LPS and IFN gamma. As I mentioned before, this converts these macrophages towards M1, or the anti-tumoral phenotype. And if you leave these with the tumor cells, these pancreatic cancer cells on top, over 48 hours to 72 hours, excuse me, and you remove the macrophages and phenotype them, what you start to see is their phenotype changes. So initially, when you have the M1 stimulated macrophages, these will be producing TNF, producing IL-1 beta. These are all pro-inflammatory cytokines. So you see the untreated cells have a very high, robust pro-inflammatory phenotype. As you start to co-culture them, you start to see the shift. And as they are cultured for 48 hours, for example, even though you started with M1, now they're starting to see this M2 phenotype. So it means that something in these cancer cells through EVs are reprogramming these macrophages towards the M2 phenotype. And then if you keep it for 72 hours, again, with these M1 type macrophages, you continue to see increase in these arginase and IL-10, which are both anti-inflammatory phenotypes, anti-inflammatory markers, or pro-fibrotic markers. So arginase is a very common anti-inflammatory, and the higher values that you're seeing at longer 48 and 72 hours shows that these previously M1 macrophages, by basically just this co-incubation with the pancreatic cancer cells, have been repolarized over that 48 hours. And we saw that exosomes are the ones that are doing it. We collected these exosomes, these electron microscope of the exosomes collected through the media here. And then more importantly, if we had our pancreatic cancer cells that were luciferase expressing, we could actually measure the luciferase signal in macrophages over 48 hours and 72 hours, which we saw, we quantified in this graph below. So through this, we were able to show that pancreatic cancer cells, human pancreatic cancer cells, have the ability to transfer these pro-inflammatory macrophages towards an anti-inflammatory or pro-tumoral phenotype based on the content that was transferred from the pancreatic cancer cells to these macrophages. Then Meiju decided to ask the second question: if we were to if we were to transfect these pancreatic cancer cells on the top of that cell culture insert with specific microRNA producing plasmid DNA, and as I mentioned, microRNA 155 and 125B are the ones that are producing this M1 phenotype from M2. And now you are enriching the EVs with these microRNA 155 and 125B. Could you then change this M2 phenotype macrophages towards M1? And so she did that transfection experiment. These are HAPI particles that then encapsulated the DNA. We have 3 different types of plasmid, one that encodes for microRNA 125B, and another one that encodes for microRNA 155, and then a null vector as a negative control. So this is a null plasmid, 155B expressing plasmid, and then 155 expressing plasmid, and 125 expressing plasmid. And first, she characterized those plasmids with restricted digestion to show that the cDNA was correct, and that we were actually using the right type of vector system. And she also transfected using our HAPI in these pancreatic cancer cells, and then by PCR showed that in the transfected cell cytoplasm, there was a huge increase in microRNA 155 expression, as well as a huge increase in microRNA 125B expression. And when she collected EVs, she was able to also see upregulation of these microRNAs in the EVs from pancreatic cancer cells, both for microRNA 155 and 125B. So we knew that these microRNAs were overexpressed in pancreatic cancer cells, could be secreted in the EVs as well, and they will be affecting this transfer into macrophages once we start to do this co-culture experiment. And that's what she did next. She did 2 experiments. The first one, she took out the EVs from the cell culture media and then fed them to the macrophages, so that we want to make sure that it is EV-mediated communication, and not just have the macrophage sit on the pancreatic cancer cells sit on top of the macrophages, because there may be some other factors that are also going from that cell that would mask the effect. So in this case, what you're seeing here is we have our macrophages that are untreated. We first stimulate them with M2 using IL-4. So now we're not relying on that tumor cells themselves to change them to M2, but we are now actually artificially creating M2 phenotype. And then we have these transfected PANC1 cells sitting on top that were basically allowed to sit for 48 hours. And then 2 days later, we collect the J77 cells from the bottom and start to see what is going on in terms of the different markers of M1 to M2. And just to remind you, IL-1 beta is an M1 marker, arginase is an M2 marker. And so what you're looking at figure A is just IL-1 beta, and you see that when you have these macrophages sitting with these transfected IL-155 or 125B transfected PANC1 cells, you start to see an increase in IL-1 beta, which is an M1 marker or a pro-inflammatory marker. Similarly, you see a decrease in Rh1, which is characterized by a decrease in M2 phenotype. If you take the ratio of the two, you clearly see an increase in M1 to M2 ratio. That's this C here. And then similarly, so the top is the combination. And then in the bottom here, D, E, and F are also looking at the other markers. Inos is one, which is increased, which is an M1 marker. Arginase is an M2 marker. And what you're seeing here is a ratio of the two. And so by just allowing these PANC1 cells to sit on top and the macrophages to sit at the bottom, we see EV-mediated communication that sends a different message to the PANC1 cell, to the macrophages, and now they have been able to convert them into an anti-tumoral phenotype based on the fact that we have over-transfected microRNA 155 and 125B in our pancreatic cancer cells. So the message from this work is that if you were to create transfection in these cancer cells and subsequently look to see EV-mediated propagation of the message, it's almost like you're looking at tweets that are secreted from these cancer cells that are affecting communication in the microenvironment. And if those tweets could be hacked in a way that you are now changes the message, and now you have basically made that message to be anti-tumoral as opposed to pro-tumoral. So you're using EVs, but with a different message inside, specifically these microRNAs that are overexpressing and transferring over to give this anti-tumoral phenotype. Similarly, we did the second study, very much like the first one, but in this case, not necessarily having the cells sit on top of each other, but take out the EVs. And so she went and purified the EVs by differential centrifugation and gel filtration, and then treated the macrophages with the EVs or the enriched EVs that had these microRNAs 155 and 125B, and again showed the same sort of properties that if you increase, so again, we are looking at IL-1 beta and Rh1 in these two and the ratios of those two markers on the top, and then Inos, arginase, in the ratios of those two in the bottom. And what I want to focus you is just the last part here, where we are, after we treat, we are seeing a decrease in this M1 marker is increased, M2 marker is decreased. Again, if you take the EVs that are enriched in these microRNA 155 and 125B, so this is truly an EV-mediated phenomena, and subsequently we know how to transfer these EVs and control this phenotype very well. But this is done at a cell level. Could we actually start to think about how can we do in vivo? So Neha was a postdoc in my lab. She's now at Thermo Fisher in San Diego, and we collaborate on this project with Dr. McKenzie, who is at University of California, Davis. Dr. McKenzie has a pancreatic cancer genetically engineered mouse model. And this mouse model is developed in C57 black mice. And so these are immunocompetent. We can look at macrophages in this model, and the tumor grows in the pancreas of these animals. So we created a hyaluronic acid nanoparticle specifically to target M2 macrophages in vivo using a peptide. This peptide was first identified in a lab at University of Washington in Seattle. It's based on phage display technology that targets specifically M2 phenotype of macrophages. We added 3 glycine spacer and a cysteine to create a thiol group at the end so that we can form a conjugate with our PEI, so that, with a PEG, so that when we attach this peptide, it is tethered by a PEG chain, a 2000 molecular weight PEG chain. And this just shows the NMR and particle size. We are able to encapsulate microRNAs. And what I will be showing you is the data with microRNA 125B given in vivo. So the next question we asked is, how do we get to macrophages in the pancreatic tumor in vivo? And interestingly, what we have shown in the past and continue to show that if you take mice and inject any formulation, and especially our HA particles intraperitoneally, the peritoneal macrophages are transfected by that particular type of nucleic acid molecule, in this case, microRNA 125B. So what we showed that peritoneal macrophages, especially the large peritoneal macrophages that are F480 positive, will pick up our nanoparticles, and you see this co-localization after about 6 to 12 hours post-administration of the dose. But more importantly, if you inject microRNA 125B, and now you're able to change that phenotype in the peritoneal cavity towards a anti-tumoral or M1 phenotype. And you see this by increasing in the iNOS to arginase ratio, as well as the fact that microRNA 125B is overexpressed in these macrophages. So we're doing this all in the peritoneal cavity of a mouse. And subsequently, what we have found is that these peritoneal cavity macrophages migrate to the tumor. And that's what we showed with the, this is in the GEM model, the KPC genetically engineered mouse model of PDAC with Dr. McKenzie, that our nanoparticles injected in the peritoneal cavity, specifically with this M2 peptide, were able to reach the pancreas of these tumor-bearing mice. And subsequently, once they reach the pancreas, you see there's also some liver accumulation, but it doesn't go to spleen or kidney. There's a very, very nice enrichment in the pancreas. So with this data, we said, the peritoneal macrophages have this tropism towards the disease phenotype. Then can they actually go and to convert these macrophages in vivo into the anti-tumoral phenotype that we want? So what you're seeing is that the orange, the light orange color here is the M2 macrophages, M2 peptide targeting. In the red is the scrambled microRNA. So it does have the same peptide, but the microRNA is non-functional. And you see that in all cases, CD86 is overexpressed, but CD206, which is the M2 marker, is downregulated. And that clearly shows you that we have been able to convert these in vivo into this anti-tumoral phenotype. Now, how does EVs get involved in this? Well, we see in many of these instances that when peritoneal macrophages are able to go to those distal sites, they will change the microenvironment and ultimately create this anti-tumoral phenotype. But in order to enhance the transfection efficiency in vivo, EVs are actually creating a bystander effect. So they continue to propagate the same way that we are able to control the message in vitro. In vivo too, we are seeing many examples where EVs are expanding the transfection ability once once you're able to transfect with our nanoparticle system using hyaluronic acid, we can then rely on EVs to actually propagate that message. So clearly, even in vivo, we are seeing some very beneficial properties of EVs in terms of enhancing the transfection ability. And I'll show you additional data in the next example, how EVs can be a really important way to enhance the transfection efficiency and create that polarity that we desire. So moving into the next example, we are focusing on ovarian cancer. So again, we are interested in a lot of these solid tumors that are very difficult to treat and ultimately create a very high mortality rate. And in ovarian cancer specifically, we are interested in looking at, so women who are diagnosed with ovarian cancer usually occur at late stage again, but they have this adjuvant, so first surgery followed by adjuvant chemotherapy, and they undergo this remission phase. And typically this remission phase could last for up to about a year, maybe a year and a half. But in many instances, there is this relapse or recurrence that occur. And then the disease that occurs at recurrence, almost 80% of the advanced stage patients have this recurrent disease. And this recurrent disease is resistant to chemotherapy. Whatever chemotherapy was tried in the initial stage fails, and patients subsequently has a much worse prognosis, and majority of those patients will die relatively quickly. So our question is, what happens during this recurrent stage or relapse stage? How can we understand the biology a little bit better? And subsequently, what can we do to affect therapy, especially for these patients who are relapsed? So Shrujan Gandham, was at Moderna here and is just recently transferred to another company called Era Therapeutics, was a PhD student in my lab. And he came in and we developed specifically a resistant model, a relapse model of an orthotopic ovarian cancer. And this is also immunocompetent so that we can look at the immune system in this model. The way we designed this model was we took the female black mice, we administered the ID8 VEGF murine ovarian cancer cell line. So these are cells that are of ovarian cancer origin from mouse, but they are stably transfected with vascular endothelial growth factor, so they're pro-angiogenic. And these cells are injected in intraperitoneally, so they cover the peritoneal bed. And then after 14 days, we start them with chemotherapy. So we start to see that once the tumor burden starts to grow, the animals start to develop ascitic fluid, very similar to what happens in women with ovarian cancer. And then we start treating them with chemotherapy. So we give 3 doses of 8 milligram per kilogram just to match the maximum tolerated dose IP. And then we stop therapy at day 28. And what you see here is that the animals that are treated with paclitaxel initially do not have tumors, and their body weight remains constant, but then they start to develop tumors. And so what you see is ascitic fluid buildup again because of this relapse disease, the disease that recurs. We also have another group where we have given the tumor, but we do not give them any treatment, just leave them to have the continuous tumor. And then we sacrifice both groups at day 56 or day 60 approximately, and we collect tumor nodules from the peritoneal cavity, from the diaphragm, which is the metastatic disease, from the omentum, from other parts of the peritoneal cavity, as well as collect ascitic fluid, and start to look at the genotypic properties of these tumors. What is the actual characteristics of the tumors that have not been treated, that you see in the blue here, versus those that are treated and now have relapsed, that you see in the orange? And what are the differences in the genetic signatures between the two? And why is it that these ones become resistant to chemotherapy once you have treatment and then relapse? So what we found was very interesting. We found that once you collect these tumors, either they're primary tumors growing in the peritoneal bed or in the metastatic nodules that are growing in the diaphragm, all of them, if you look at the ones that had been treated and then relapsed, are resistant, and they have all of these resistant markers such as the efflux transporters genes, ABCB1, multi-drug resistant protein 2 are overexpressed. So you see a lot of these genes overexpressed relative to the control, those animals where the drug was not given at all. So we are comparing the paclitaxel treated versus the control, and you see overexpression of all of these resistant genes. Similarly, in the metastatic nodules, you see this very large increase in the resistant marker. But what was interesting is that when we start comparing with the control animals that did not receive any treatment to those that received paclitaxel and then relapsed, we found several microRNAs that were either downregulated or upregulated. And one particular one that caught our attention was microRNA LET7B, which is a tumor suppressor microRNA. And that was shown to be downregulated by about almost 4.5 to 5-fold from those that in the paclitaxel treated group that relapsed versus the ones that the tumors were not treated with any drug. And this LET7B has been well known to correlate with clinical prognosis of pancreatic cancer. If you look at patients, so LET7B targets this HMGA2 protein expression in pancreatic cancer. And what has been shown is that women who have low levels of LET7B tend to do poorly in prognosis of pancreatic cancer, and they tend to have much higher mortality rate. Similarly, when you look at the ratio of expression to the mortality, LET7 expression and HMG expression, you see that there's a very strong correlation in the clinical setting as to that these women will do much worse in their prognosis. So we asked a very simple question: could we administer exogenous LET7B into mice that are resistant to these or have relapsed? And subsequently, what will be the effect when you combine it with a chemotherapy drug? How would that help in terms of treatment? And we found really interesting work. So we again, our paradigm is to create the tumor model, treat it with 3 doses of paclitaxel, induce that relapse, and then start to look at whether we can treat it with LET7B and paclitaxel combination, and see how is that different from just paclitaxel alone in terms of therapy. And what we found is that we can completely reverse the resistant phenotype in cells when you are combining it with LET7B. You can clearly see the naive cells, they die at very low doses of paclitaxel. With this combination, we have been able to suppress mostly, almost around almost tenfold down in terms of the doses needed, and statistically significant compared to scramble sequence of LET7B. But more importantly, all of the genes that previously were upregulated, these resistant genes, have also been downregulated because of this exogenous LET7B administration. So this is at the cell level. We did the same thing in vivo, and we took the tumor, we inoculated, again, created that resistant phenotype with 3 doses of paclitaxel, gave LET7B intraperitoneally at 3 doses, then treated with paclitaxel, and then at day 55 or around day 60, euthanized. When you look at body weight, these animals did not have ascitic fluid at all, so they never gained body weight. The change in body weight is almost back to baseline. But one of the key hallmark of our tumors is VEGF production. See this huge increase in VEGF once you start to induce tumors. That came back to baseline with this combination treatment in this relapse model, and you can clearly see this in these pictures of mice abdomen. But this was a huge decrease in VEGF corresponding to the fact that when we administer LET7B into these animals that have relapsed ovarian cancer, they actually are able to get that sensitivity back to paclitaxel, and they respond much better to paclitaxel, and they are able to get much safer therapy. So we will ask, is this just because our HA particles are good, or is this something else happening in vivo? And we interestingly found that if you collect ascitic fluid from these animals, whether they're controlled or treated, we could harvest EVs from the ascitic fluid. And even 48 hours post-administration of our LET7B intraperitoneally, we were finding about 7.5-fold increase in the secretion of LET7B in the EVs, these extracellular vesicles in the ascitic fluid. Now, we don't know which cell has produced these. We assume that they are tumor cells, but it's possible that other cells have also picked up these LET7B transfection myeloid cells, as well as because these HA particles are non-specific, they will go to a variety of different types of cells and transfect. But we are seeing this incredibly robust increase in LET7B in the EVs 48 hours after administration. The scramble obviously doesn't do anything, and then this is just the control animal. So our hypothesis here is that if we were to administer our HAPI nanoparticles with any microRNA, it will transfect a few cells. But then those transfected cells can then help us in terms of increasing the propagation of our transfection through EVs. And the analogy, here in Boston, we have a problem with traffic. We have a very, very dense, there's a joke that it takes, if you are here in Boston for 2 hours driving, you will still be in Boston because the traffic is so heavy, especially in the morning, in the rush hour time period. So if we were to think of delivery as a problem where there's a high density of traffic, and ultimately, our nanoparticles will take a tortuous path, our hypothesis is that if we could deliver to a few places with our HA particle, but then if use EVs like bicycles, and those bicycles will be able to go through narrow streets and narrow alleyways, and they will then help us in increasing the propagation of our message even further than where our HA particle has been able to do. And so here you see a picture of Boston's mayor actually advocating for the fact that we should be using more bicycles in this city as opposed to driving cars, just so that it's a better traffic and less pollution. But in terms of EVs, we see a huge potential in vivo of EVs helping us to basically take these HA particle transfected cells, use the EVs from those cells to help us in spreading the message even further. So to even further characterize this, Srujan went ahead and did a modeling study, an in vitro modeling study, again, using these transwell plates. And what he did was he put these cancer cells, these ID8 VEGF cells with let7b transfection, and then use untransfected cells at the bottom and tried to characterize these EV-mediated propagation based on the donor cells being on top and the recipient cells in the bottom, to see, and then collected EVs from these fractions. And so he wanted to see how many layers of cells could you actually reach ultimately with one single transfection. So if you had one donor cell, how many potential recipient cell layers can you go after? And we saw about 4 layers of cells could receive these EV-made let7b that was secreted in the EV and ultimately allow for transfection. He was able to model that very well. So the red line is the actual predicted bit based on his model, and the blue line is what was observed experimentally. And because we know that let7b target is HMGA1 that I mentioned before, this HMGA2, so we collected the HMGA2 mRNA from these cells that have been transfected, and we were able to do pharmacodynamic response. And so we're able to do this, this calculation of dose response relationship. So if we had this many copies of microRNA, what would be the ultimate response at a cell level? And we showed that in any given layer of these cells, for EC50 of these microRNA let7, we needed 374 copies of the molecule. And that's what we could get up to 4 layers deep from a single transfection. If you want to go to EC90, we needed about 471 copies of the molecule per cell to be able to get that 90% response in HMGA2 mRNA expression. So these are the kinds of studies that we are continuing to do, but we see great potential of EVs in terms of propagating the message and ultimately helping us create a better therapeutic strategy, especially with these microRNAs and modulating the tumor microenvironment. We don't use EV specifically for drug delivery because, to me personally, harvesting EVs and loading them outside with drugs is just not a practical way. We have much better synthetic delivery systems, and I feel that EVs would not be as efficient, and more importantly, the yields are very low. But if you can use EV in vivo to actually convert the message that you want to go to these neighboring cells, that is a much better strategy, in my opinion. So I hope I give you enough in this short presentation to basically tell you a little bit about EVs and how EVs are critical communicators in the context of sustaining tumor growth, angiogenesis, and metastasis. We are very interested in evaluating the roles of EVs, especially in microRNA-mediated tumor reprogramming and looking at therapeutic outcomes, whether it's immunosuppression or in the context of resistance and relapse. And so I gave 2 examples. The first one was on pancreatic cancer, where I showed specifically that we can repolarize these tumor-associated macrophages based on the communication that happens between tumor cells and EV-mediated microRNA transfer into macrophages or myeloid cells. And then the second example, specifically, how do we control for microRNA delivery into these relapse and resistant tumors, such as ovarian cancer, by increasing the propagation and creating the bystander effect. So I hope that in both the examples, I have convinced you that first, it's important to understand tumor biology, but clearly, EVs have a huge role to play and could have a huge effect in terms of enhancing therapeutic benefit in these hard-to-treat tumors in patients, especially ovarian cancer that's relapsed or pancreatic cancer. So thank you again for your attention this morning or this afternoon, wherever you are. I'm going to stop sharing so I can see all of you and hopefully answer your questions.
Thank you very much, Professor Amiji. It was very fantastic and very interesting talk. Especially many of us are working on EVs, but not many of us working on cancers, but it's a kind of eye-opening for us as well. So do we have any questions from the audience? Or even you can send them in the chat box, then we can see it as well.
Yeah, either just turn your camera on, or you can just
Yeah.
put it in the chat. I'd be happy to answer any questions.
Yeah.
I have a question actually.
Yeah, please go ahead, get that. Yeah, you can.
My question is like this microRNAs are negatively charged, and HA, hyaluronic, is already negatively charged, and you use PEG, right, to connect it?
No, we use, so yeah, hyaluronic acid is negatively charged. MicroRNA is also negatively charged, but polyethylamine is positively charged. So if you covalently link polyethylamine to the HA backbone, you're able to create these what are called comb structures. So they're like, they have these side chains of PEI, polyethylamine, and that gives the positive charge. Now, you don't want too much of polyethylamine because it's going to create toxicity. So we control the density, but then we also reduce that charge density with HA with polyethylene glycol. And now what you're able to do is you have a way to get encapsulation. You have a particle that has the PEG chain on the surface, HA on the surface, but the nucleic acid is in the core. And then you go through this exercise I mentioned before, where you test whether it's stably encapsulating, is it delivering into the cell, is it based on that CD44 expressing receptor and endocytosis, is it getting out of the endosome, is it transfecting, and you get a very, very good structure functional relationship. But it's really the PI that's helping us in encapsulating the microRNA.
Thank you. But in that case, this is a very multi-layered nanoparticle with different target. But my question is like, how does it go to the target point? Because you said cysteine, this disulfide bond formation will be there and to be broken. What caused this disulfide bond to be broken? And this delivery goes to our target.
Yeah, so there are many ways that you can, in the examples I provided, like for example, in the pancreatic cancer, we know that the cells, these macrophages express CD44. And so there's CD44 and HA binds to CD44. So that's one way. We also put this M2 specific peptide. So that targets the macrophages that are M2 and allow us to get into these macrophages as well. The other way to enhance the delivery efficiency is so yeah, you can you look at CD44 expression as one way to target, but your question about the disulfide specifically is not just to target, but once it gets into the cell. So you want this package to be fairly stable in blood. You want them to be fairly stable in the ascitic fluid or in the peritoneal fluid. But you want it to release the cargo when it's in the cytoplasm. So, and there is about 100-fold difference in glutathione level between the plasma or ascitic fluid and intracellular compartment. So intracellular compartment has a higher glutathione level compared to extracellular. So what happens is if you create disulfide crosslinks, these disulfide crosslinks are broken by glutathione. So your package unfolds itself, and now you're able to release the nucleic acid better in the cytoplasm of the cell compared to outside or where you are in the blood or in the ascitic fluid. So that's why we use disulfide bonds and try to create the particle where you have this cross-linking mechanism. But generally for targeting, HA itself has intrinsic targeting ability. We can decorate these particles with different types of peptides. We have done EGFR. We have done these M2 macrophages. You can also put folic acid and target folic acid expressing cells. So there are a number of ways that you can target specific types of cells based on receptors that are overexpressed on those cells compared to healthy cells.
Thank you. Can I ask Suranga?
Yeah, go ahead, please. Yeah.
That's other ones like this. What we see so far are synthetic nanoparticles, and these synthetic nanoparticles, they have high immunogenicity and they also high rapid, clearance from our blood vessels or system. Why can't you use like EVs, which are more natural origin? Have you thought about?
So I shy away from like taking EVs out and then filling them with drugs. And the reason why that is because if you look at, there are two problems right now with EVs. Even, if you look at companies that have invested so much on EVs, one of them was called Kodiak, which, filed for bankruptcy here in Cambridge, where they're trying to do drug delivery. And the problem is EVs, even if you're trying to use single cell line, like they were using, these epithelial kidney cells, HEK, and they're culturing it and then producing large amounts of EV, and they had a very well characterized system, GMP grade material. They were still finding a lot of heterogeneity in EV. So every time you make EV, there's a batch to batch differences. And then second, in both in terms of surface composition and content. And then if you are to fill them and put your drug in the EV, what happens is they change the properties. So when you inject those EVs back into the animal, they're no longer naturally derived EVs. They are going to be synthetic systems, and then they get picked up by liver and spleen, and they're not going to your target. So our approach is not necessarily to take EVs and fill them outside, but can we use our synthetic particle, direct them to the cells, to a few cells in vivo, and then let those cells then produce EVs themselves and expand the delivery efficiency through EV inside the body, and do that. So you have much better control of the EV production and continue to propagate that message, because now you're doing everything inside the body as opposed to taking it out, filling it, and then putting it back, because we just don't think that system is efficient enough. And will give you, especially translatable, if you can go with that system towards the clinic. But this way, you will utilize well-characterized synthetic systems, transfect, and then be able to then utilize that and you can still get the benefit of EV in vivo.
Okay, thank you.
Sure.
Are there any other questions? May I ask one simple question? So it's known that almost all these EVs will be ended up if you inject it from outside, mostly in the liver. So how about these nanoparticles that you use? Do they have the same tendency like getting accumulated in the liver as well?
Yeah, so usually, if you look at what has in the field generally, you are going to get some, and you saw even in our pancreatic cancer example, liver uptake, especially when you administer. So liver is going to take up the question. There are two ways that you can influence therapy distal from the liver. One is if you can design these particles or make these cells be more tropic towards the disease area. So in this case, if you get enough density in pancreas to effectively treat the disease, then even and whatever goes to the liver, assuming that it doesn't provide any safety concerns, you are still in a pretty good shape. So you get that risk to benefit. The other way to think about is targets. What are your targets? How are the targets expressed in your disease tissue compared to liver? If you can show that, like, for example, these our approach is microRNA 125B, and we have these M2 macrophages only in the pancreas because that's where the tumor is, or in the metastatic disease elsewhere, but not necessarily, may not be as heavily populated in the liver, then that target is also present only in that area. So even if you get microRNA in the liver, it's not engaging with the target there. So that's how you are able to get benefit and improve the efficacy of your therapy. But you in this delivery system, whether you give EVs from the outside or nanoparticles, liver and some of these organs of the reticular endothelial system will still be important in picking the particles.
Okay. Do we have any other questions from the audience? In the absence of any questions, I would like to thank again very much for having the lecture with us today and spending your time with us with this, your data and all the insight that you shared. So on behalf of the Combivet and OH Boost and on behalf of the EMU, I would like to thank you again. And yeah, so if there's anything, I think we would like to be in contact with you, especially the expertise on drug delivery. I think in the future, I think it would be very beneficial for us as well. Thank you very much, Professor Amiji. We really appreciate it.
Thank you. Pleasure to be with you this morning and this afternoon for you guys. Yeah, any questions that if you comes up,
Yes.
you know,
Yes,
at
we
the
are
time.
also very much interested on drug loading and drug delivery, all this, you know, EV release and EV uptake, tropism, all this actually, you know, burning questions among ourselves. Probably, yeah. We'll
Yeah,
be keeping touch with
happy
you
to,
as
you
well.
know, find some collaboration. So thank you.
Yes.
Yeah,
Thank
have a
you.
wonderful day. Bye.
You too. Thank you very much. Bye-bye.